The hypothesis that fruit peel nutrition varies dramatically with species, cultivar, and growing conditions is both compelling and strongly supported by emerging research, revealing how the intersection of genetics and environment shapes the nutrient density of peels. Understanding these variations is critical for fully harnessing peels as functional food ingredients, especially when comparing tropical and temperate fruits. One striking example is the difference in anthocyanins, powerful antioxidant pigments, between red and green-skinned apple cultivars. Red apple peels accumulate cyanidin-based anthocyanins, contributing both vibrant color and enhanced antioxidant capacity, while green varieties largely lack these compounds despite comparable levels of other nutrients like quercetin. This illustrates how genetic differences within a single species directly determine peel bioactive profiles, suggesting that cultivar choice alone can double or triple certain peel-derived antioxidants. Tropical fruit peels show even more pronounced nutrient richness. For example, mango and papaya peels contain abundant carotenoids, SS-carotene, lutein, zeaxanthin, that are often negligible in temperate fruit peels. These carotenoids are critical for vision, immune health, and inflammation modulation, and they accumulate more in peels of fruits grown under intense tropical sunlight. By contrast, peels of temperate fruits like pears or apples rely more on flavonols, quercetin, camphorol, and phenolic acids for UV protection and defense, resulting in a different polyphenol spectrum. Environmental effects compound these genetic differences. Multiple studies on citrus grown in tropical versus subtropical regions show peels from tropical grown fruits often contain higher vitamin C and phenolic levels, likely reflecting increased plant stress responses to stronger sunlight, higher humidity, and more variable rainfall. Similarly, grape cultivars grown in Mediterranean-like climates accumulate more resveratrol in their peels compared to the same cultivars grown in cooler regions, underscoring how climatic stress can drive phenolic enrichment. Seasonal timing adds another layer of complexity. Early season fruits harvested before full maturity often have peels with higher phenolic concentrations, part of the plant's defense system that declines as fruits ripen and soften. Conversely, late-season harvests can sometimes yield peels richer in sugars and carotenoids but lower in polyphenol, depending on fruit type and environmental factors. These dynamics suggest precise harvest timing could optimize peel nutrient density, especially for functional food production. A rigorous research approach would involve systematic comparative analyses of vitamin, carotenoid, anthocyanin, and polyphenol profiles in peels of identical fruit species or cultivars grown under different climatic zones, using standardized assays like HPLC for accurate quantification. For instance, comparing peels of oranges grown in subtropical Florida to those from tropical Thailand could reveal environmental influences on peel vitamin C and flavonoid content, while side-by-side trials of red versus green apple cultivars across regions could tease apart genetic from environmental contributions. These studies could extend to exploring microclimate effects, such as altitude, soil mineral composition, and irrigation practices, on peel nutrient profiles, as such factors significantly alter plant stress levels and secondary metabolite production. This direction would not only clarify which growing conditions favor nutrient-rich peels, but also inform precision agriculture and breeding programs aiming to select or develop cultivars with inherently higher peel nutrient density. The outcomes of this research have practical implications far beyond academic curiosity. They could guide farmers, processors, and food manufacturers in prioritizing specific fruit varieties or sourcing regions to maximize the nutritional value of peel-based functional ingredients. They could also inform breeders to develop cultivars selected specifically for superior peel nutrition, akin to how some grape cultivars are bred for higher peel polyphenol in winemaking. Ultimately, comprehensive comparative research into varietal, seasonal, and environmental impacts on peel nutrition would fill a critical knowledge gap, enabling more consistent, nutrient-dense, and sustainable use of fruit peels. Such insights would directly advance the goal of valorizing peels across tropical and temperate fruits, ensuring functional food innovations and peel-derived supplements are grounded in the best possible raw materials for health and sustainability.